Cell biology, a branch of biology which deals with study of cell details. When was cell a fundamental unit of an organism? So in simple words, it can be defined as basic functional unit of life, that is cell. Cell term is derived from Latin word, and this cell was first observed by a scientist named as Robert Hooke in 1665. We know all the living organisms, they are composed of one or many cells to perform their individual functions. Cell is smallest unit of a life which is able to control and perform several functions in all living organisms. All unicellular organisms, including bacteria, are composed of single cell. All multicellular organisms, we can include human in this category, which are composed of many cells, and these cells are eukaryotic. So in brief, one can say that biological science which deals with study of structure, function, molecular organization, growth and reproduction, and genetic of the cell is cytology or cell biology. While looking towards the history of cell biology, one can go back in ancient time. Greek philosopher Aristotle and Paracelsus, they concluded that all animals and plants are constituted of few elements, and these elements are in repeated fashion over there. After invention of magnifying lenses, the world of microscopic dimensions was discovered. Development of cell biology associated with development of optical lenses and combination of these lenses in the construction of compound microscope. Invention of microscope and gradual improvement of this microscope makes many developments in this field, that is cell biology. Compound microscope was invented by Barnes Johnson and James Johnson. A microscope they invented had two lenses, and total magnifying power was between 10x and 30x. Later, Italian microanatomist Marcello Malpighi used microscope to examine and describe animal tissues and organs like brain, liver, kidney, spleen, tongue, lungs, etc. Dutch microscopist Anton van Leeuwenhoek succeeded greatly in improving the art of polishing lenses of short focal length. Leeuwenhoek first observed free-living cells from rainwater. And he did sketches of Bacilli, Cocci, Spirilla, and Protozoa, Hydra, etc. Leeuwenhoek was first to describe sperm cells of humans, dogs, rabbits, frog, fish, etc. In 19th century, many cell biological inventions and formulations of various theories like cell theory or protoplasm theory was witnessed by science. Mirbel stated that all plant tissues were composed of cells. French biologist Dutrochet correctly concluded that all animal and plant tissues were aggregates of globular cells. English botanist Robert Brown discovered and named nucleus in the cells of the plant like Tradescantia. After looking towards history, now we will go towards how cell measurement is done. Cells, as we know, are very small, invisible to human eye. So, the researchers or the students who study the biology must know how cell measurement is done. Micrometer was the term used formerly for measurement which equals to 10 raised to minus 6 meter. Second unit, that is nanometer, which is equal to 10 raised to minus 9, was also in use. At present, some of the units of measurements of cells are given here. 1 centimeter equals to 10 raised to minus 2 meter. 1 millimeter equals to 10 raised to minus 3 meter. 1 micrometer equals to 10 raised to minus 6 meter, and like that. So these are some of the units of measurement of cell. Now we will study types of cells: prokaryotic cell and eukaryotic cell. So in the beginning, the primitive type of cell, that is prokaryotic cell. Prokaryotic cell is simpler and smaller than that of eukaryotic cell. So here the diagrammatic sketch of one of the bacterium, we can see prokaryote. This term is derived from Greek word prokaryote, means before nuclei. Prokaryotic cell exhibit all the signs of life. They are smallest types of cells, averaging 2 to 5 micrometer in length, which makes them just visible under light microscope. Despite their size, inside each cell there is complete chemical and biochemical machinery necessary for growth, reproduction, and acquisition and utilization of energy. Prokaryotes have large array of abilities. Some of them live in absence of oxygen, some in extreme conditions of heat or cold. Others can grow at the bottom of oceans, where only source of energy is hot hydrogen sulfide bubbling up from the core of the earth. Prokaryotic cells, to which we call as a primitive cells, examples are bacteria, or blue-green algae. Prokaryotic cell consists of central nuclear component, which includes DNA, RNA molecules, and nuclear proteins, surrounded by cytoplasmic ground substance. Which is enveloped by plasma membrane. Prokaryotes do not have nuclear apparatus nor have respiratory enzyme systems, which are separately encoded in membranes. Although inner surface of plasma membrane itself may serve for enzyme attachment. Cytoplasm of prokaryotic cell lacks well-defined cytoplasmic organelles like endoplasmic reticulum, Golgi apparatus, mitochondria, centrioles. Also, they don't have nucleoli and basal bodies. Now, shape of the cell. Basic shape of eukaryotic cell is spherical. However, shape is ultimately determined by specific function of cell. Thus, shape of cell may be variable, that is frequently changing, or may be fixed. Variable shape occur in amoeba and white blood cells. Fixed shape of cell occurs in almost all protists, plants and animals. In unicellular organisms, cell shape is maintained by tough plasma membrane and exoskeleton. In multicellular organism, shape of cell depends mainly on its functional adaptations and partly on surface tension, viscosity of the protoplasm, cytoskeleton of microtubules, microfilaments and intermediate filaments. The mechanical action exerted by adjoining cells and rigidity of the plasma membrane is also responsible for determining the shape of the cell. Cell shape may vary from animal to animal and from organ to organ even. Even the cells of same organ may display variations in shape. Thus, cells may have diverse shapes like polyhedral, cuboidal, discoidal, spherical, spindle shaped, or elongated, or branched. Among plants, cell shape also depends on function of the cell. Cell size. Eukaryotic cells are larger than that of the prokaryotic cells. Eukaryotic cell size range between 10 to 100 micron as compared to prokaryotic cell size, which ranges between 1 to 10 micrometer. Size of cells of unicellular organisms is larger than a typical multicellular organism cells. For example, amoeba proteus is the biggest among unicellular organisms. Its length is 1 micrometer. One species of Euglena has length up to 500 micrometer. Single-celled alga, Acetiobularia, which consists of stalk and cap, is very large in size and measures up to 10 centimeter in height. Multicellular organism cell size ranges between 20 to 30 meter. Largest animal cell is the egg of ostrich, which has 18 centimeter diameter. Cell volume. volume of cell is fairly constant for particular cell type and is independent of the size of the organism, and this is called law of constant volume. For an example, kidney or liver cells measures about the same size in bull, horse and mouse. The difference in the total mass of organ or organism depends on number and not on volume of the cell. Thus, cells of elephant are not necessarily larger than those of other tiny animals or plants. The large size of elephant is due to larger number of cells present in its body. Cell number. So number of cells present in the organism, it varies from single cell in unicellular organism, for example protozoa, to many cells in multicellular organisms, for example plants or animals. Number of cells in multicellular organism usually remains correlated with size of the organism, and therefore small sized organism has less number of cells as compared to large sized organisms. If we take example of human being weighing 80 kg may contain about 60,000 billion cells in his body. Cell biology and other biological sciences. Cell biology, no doubt, is helping and had helped to understand various complicated life activities like metabolism, differentiation, heredity, growth, evolution, and also its study is very important to make us familiar with what happens at cellular and molecular levels. Due to this wide application in various branches of biological sciences, cell biology plays a role. Some of them are as follows: cytotaxonomy or cytology and taxonomy. We know each plant and animal species has definite number of chromosomes in its cells, and the chromosomes of individuals of species resemble closely with one another in its shape and size also. These characteristics of chromosome help taxonomists to determine taxonomical position of the species. Further, cytotaxonomy plays important role in giving strong support to the manner of origin of certain taxonomic units. One can define cytotaxonomy as cytological science which provides cytological support to the taxonomic position of any species. Second, cytogenetics, cytology and genetics. Cytogenetics is branch of cell biology which is concerned with cytological and molecular basis of heredity, variation, mutation, phylogeny, morphogenesis and evolution of organisms. Mendel's law of inheritance and concept of gene could be well understood only after application of cytological concept to the genetics. So this is the role of this cell biology in cytogenetics. Now, cell physiology, correlation of cytology and physiology. Cell physiology is the study of life activities, size, nutrition, metabolism, growth, excitability, reproduction or cell division and differentiation of the cell. Cell physiology helps to understand various complicated physiological activities at cellular level. Cytochemistry, cytology and biochemistry. Cytochemistry deals with the chemical and physicochemical analysis of the living matter. Due to its study, presence of carbohydrates, lipids, proteins, nucleic acids, and other organic and inorganic chemical compounds in the cells can be determined, and this is of great importance. Cytopathology, cytology and pathology. So, application of molecular biology to pathological science no doubt has helped in understanding various human diseases at molecular level. Because most diseases are caused due to disorder of genetic codes in DNA molecule, which alter the synthetic process of enzymes and ultimately disturbs metabolic activities of the cell. Cytoecology, cytology and ecology. Cytoecology is science in which one can study the effect of ecological changes on the chromosome number of the cell. Cytological studies of the plants and animals have revealed that ecological habitat and geographical distribution have the correlation with chromosome numbers.